Head kuulajad, dear friends and colleagues. Täna on meil au kuulata meie järjekordset professori inauguratsiooniloengut. Ja ma lühidalt tutvustan kandidaati eesti keeles teile. I can briefly start in Estonian and then the main lecture is in English. Pérez Carmona on lõpetanud 2006. aastal Madridi Tehnikaülikooli, kus ta läbis siis nii bakalaureuse kui magistri taseme metsanduse erialal ning 2010. aastal Madridi autonoomse ülikoolis kaitses ta magistrikraadi veel kord ökoloogia erialal. Sealsamas Madridi autonoomse ülikoolis kaitses ka ta oma doktoritöö 2012. aastal ning jätkas siis järeldoktorina nii oma koduülikoolis algul ja hiljem siis Norra loodusuuringute instituudis. Aastatel 2014–2016 töötas ta teadurina Tšehhi Vabariigis České Budějovice ülikoolis ning alates 2017. aastast on ta Tartu Ülikooli töötaja, esialgu siis makroökoloogia teadurina ökoloogia ja maateaduste instituudis. 2021. aastal valiti ta kaasprofessoriks selle samal erialal ning nüüd siis aastal 2024 on ta kinnitatud funktsionaalse ökoloogia professoriks ökoloogia ja maateaduste instituudi botaanika osakonnas. Carmona on paljude kõrgelt tsiteeritud artiklite autor ning teda on tunnustatud juba lühikese aja jooksul siis mitmel moel. Nimelt on ta 24. eelmisel aastal pälvinud Euroopa Teadusnõukogu väljakujunenud teadlase grandi, mis on väga kõrge tunnustus ühele teadlasele. Ning samuti samal aastal, siis eelmisel aastal, omistati talle riigi teaduspreemia geo- ja bioteaduste alal. Ning tänane loeng on siis eesti keeles kõlaks elurikkuse tunnuse põhiline käsitlus, aga Carlos jätkab nüüd inglise keeles teile, mida see siis tähendab. So please, floor is yours.
So, thanks a lot for the introduction, and thanks everyone for being here, for organizing this very nice event. And today I'm going to talk a bit about what I do, what we do in my group, about trait-based approach to understand biodiversity, trait-based ecology. So, I am an ecologist and I study biodiversity, and biodiversity is something that can be defined as the variation among organisms. So it includes variation in many aspects, in many groups of organisms, and particularly one of the aspects in which organisms differ is their functional attributes, which is what we are going to call their traits. So today I'm going to talk to you about this diversity of traits, why I think it's important, why I think it matters, and how we try to estimate it. So, as a brief overview of what the lecture is going to be about, we will start discussing what are traits, why they are useful. Then we will talk a bit about trait spaces and functional structure, and then I will show some examples of recent research from our group and some examples of plans for the future, things we want to do in the next years. So we will start first with what traits are and why they are useful. So a trait is defined as any morphological, physiological, phenological, or behavioral feature that you can measure in an organism. So it's something quite general. To give some very simple example in a tree, here we have an example of a tree with three traits. And the first trait that we have is the height, which is defined as the distance between the ground and the highest photosynthetic tissue of the plant. Then we have another trait, which is leaf area. Is pretty much defined by its name. And then we have another trait, which is mycorrhizal status, which tells us whether the plant in the roots is associated with mycorrhizal fungi or not. And these traits, they take trait values, so we can measure them, we can estimate them. So in this example of this tree, we have that the tree is 3 meters tall, it has a leaf area of 15 square centimeters, and it has mycorrhizal fungi in the roots, so it is mycorrhizal. So an important aspect of this is that trait values within a species are not fixed, so they change in response to different things, like changes in environmental conditions, like time, changes in genetics, etc. So for example, here the image shows how the traits of our tree change along a gradient of nutrient availability, so that individuals that are individuals that are in conditions of low nutrient availability are shorter, with smaller leaves and forming mycorrhizal associations, whereas individuals under conditions of high nutrient availability are taller, they develop larger leaves, and they might survive without mycorrhizal fungi. And why are traits useful? Well, I think traits are interesting to study because of many reasons, but I'm going to mention essentially two of them today. First is because the traits of an organism directly control how this organism is adapted to the environment. Here we have different examples with plants and birds across different environments, and you can see that they are obviously different, and this different that we see is because they have different traits. And because of this, because different traits provide different adaptations to environments, we can see that different environments have different traits. And this scheme is very interesting to illustrate this idea, and it shows what we call a functional space. We will talk much more about functional space later on, but essentially it's a space in which the dimensions are traits. So we have a two-dimensional functional space with trait 1 in one dimension, trait 2 in the other dimension. And within this space, we have represented something that we could call as landscapes. And these are fitness landscape. They reflect the fitness associated to any combination of traits across an environmental gradient. And you can see that we have 3 environments, and each of these environments have a different environmental gradient. And also, in these plots, we have blue and golden lines that represent the traits of 2 different species. So essentially, a species is able to be present in an environment if its traits somewhat overlap with the local fitness peak. So in principle, traits should allow us to understand which species are present in a given site, and even how abundant, how successful the species are in these conditions. And of course, this is an oversimplification, and things are not so simple in real life, because we cannot easily map fitness. It's extremely complicated to map fitness. You have to measure, follow populations of many species across a lot of time, including species that are not present in your sites naturally. So it's kind of an impossible task. But traits are in principle much easier to measure, so we can ideally use them to try to understand the relationship between fitness and environment. So here we can say very simple example illustrating how different environmental conditions select for different trait values within and across species. So in these 2 figures, the horizontal axis is showing the position of different plant communities along a productivity gradient. So the more to the right, the more productive the soils are. And the vertical axis represents plant height. In the left side figure, we have the height of individuals of different species. Each color represents a different species. And in the right side, we have the average height of the plants present in each community. So you can see, in general, more productive conditions tend to favor taller plants, both within and across species. This was a very simple example, so you get an idea what I mean. So Using traits to predict the fate of a species is important because if we are able to understand how traits are related to the environment, this should aid us to predict what will happen with biodiversity in the future, for example, due to global warming. And the second key characteristic that make functional traits useful is that they are related to ecosystem processes. A very simple example is related to leaf traits. So litter decomposition depends strongly on the traits of the leaves that are being decomposed. So you can have very thin leaves that decompose very fast, and you can have plants in your community that have very thick leaves that take much longer to decompose. And this is important for different for the process of the composition itself, but this process regulates some services that we get from ecosystem, like fertility or carbon sequestration. So here we have another example from a paper we published a few years ago, where you can see the relationship between leaf traits, in this case, the content of nitrogen in leaves, and an ecosystem process, in this case, the rate of leaf decomposition. And traits effects on ecosystem process are important because if we are able to understand how the environment and the traits interact to control ecosystem functioning, then we will be better equipped to understand what will happen in the future with the functioning of ecosystems and the benefits that we get from well-functioning ecosystems that are essential for our livelihood. So this scheme represent what is called the response effect framework. And the key idea here is that since these processes are mediated by traits, the component of biodiversity that best inform us about how this scheme works should be related to the traits that are present in the ecosystem. And this distribution of traits is what I call functional structure. And we can use it to reframe the response effect frameworks in terms of this functional structure, in a way in which functional structure is a function of the environmental conditions, and at the same time, functional structure interact with the environment to determine how the ecosystem works. And assuming that this relationship between environment, functional structure, and ecosystem functioning are fixed, are mechanistic somehow, then understanding them is essential to be prepared for a future where the environment is going to change. If we want to predict what is going to happen with our ecosystem and the services we get from them in the future, it is very useful if we are able to understand how these relationships work. And this scheme has been termed the holy grail of ecology by some prominent researchers, and this is in part because it's a very powerful idea, but also because it is very elusive. So this way, we trait ecologists, we are a bit like these Monty Python knights from the movie, chasing our holy grail, which is to predict the fate of ecosystems, but we are not very successful in doing it. And like in the movie, this slow progress sometimes feel like encountering multiple absurd, unexpected enemies. So, although there are other enemies, I have focused my research in the last year in fighting two of the main ones. And to me, the first is that trait data is very heterogeneous and incomplete. And the second is that we are analyzing this data using tools that are too simple. And I think that one way forward for the first challenge is to define and use synthetic trait spaces that allow us to better consider the dimensionality of trait variation. And in the case of the second enemy, I have worked in developing trait probability density methods that allow us to consider the full functional structure of communities under a single unified framework. And this is how we reach the second part of this lecture, in which I'm going to present concepts, the concept of trait space and functional structure. Just notice. From here on, I will show the faces of many people who have been crucial in all the research that I am going to present. It is not much, but it's my way to thank everyone. Many people are here, some people are not. but everyone who has worked with me during my career, because I would not be here today without you, without them. And I want to apologize because there is many more people. Some are not here. Some are not in the presentations, but are here. So I apologize in advance. It's not because you are less important, but I can't fit everyone and everything in this lecture. So if somebody is not here and they want, I can buy them a drink anytime. So let's start with trait spaces. I'm going to give an example with vascular plants because things are more developed for plants than for other groups. But essentially, all I'm going to explain could be applied to any organism in which we can measure traits. And one of the problems of working with traits is that there are many potential traits that one could study. There is a handbook of measurement of traits in plant ecology that includes something, I think, more than 70 traits, okay? And it's quite incomplete. There are many more traits. So this is very nice because you can go and study traits that are very much related to the process that you are interested in, but at the same time, the issue is that different people study different traits, and then synthesis is very complicated and very slow. And more important, we cannot make good predictions using traits one by one because plants act as a whole and not based on their individual traits. So one solution to solve this problem that some authors have proposed is to synthesize this information taking advantage of the correlation between traits. So, for example, a very famous example is this study from Sandra Diaz and colleagues, where they found that the information about 6 fundamental above-ground traits related to different aspects of plant biology can be summarized in a 2-dimensional space, which is what they call the global spectrum of plant form and function. And the first dimension in this space separates tall plants with large seeds and dense stems to the right from small plants with light stems and small seeds to the left. And the second dimension separates plants with light leaves, which are very efficient in photosynthesis, but have a short lifespan and are more attractive for herbivores here on the top, from plants with thicker leaves that live longer but are not so good at photosynthesis, so that these plants towards the bottom have a slower pace of life. And the color scheme that you can see here, this fry deck, this double fry deck, represents how densely populated different parts of this space are by species. So as you can see, there are 2 hotspots, 2 areas with high concentration of species, which correspond to herbaceous species and angiosperm trees, which means there are some functional strategies and combinations of traits that have been very successful, let's say, at a global scale, and you have a lot of species that show similar combination of traits, and some other combinations of traits that, for some reason or another, in the present time, are not so widely or not so common, let's say. It's not that they are not successful, but they are not so common. And this global spectrum was a very nice achievement, but it was missing below-ground traits, despite the roots are a fundamental part of plants, right? So a few years later, Joanna Bergman and colleagues used a similar approach and described a similar space for fine roots. And fine roots are particularly important because they are in charge of acquiring water and nutrients from the soil. And they describe that the functional space of fine roots is also two-dimensional. The first dimension separates plants with thick roots that depend on associations with fungi to get soil resources from plants with thin roots to the left that are able to explore the soil more efficiently on their own. And the second dimension is root economic gradients, which is similar to the one for leaves. And well, so, of course, after this root economic space paper was published, we saw a very good opportunity to put these 2 databases together and try to create a whole plant space, which is what we are calling now the unified plant functional space. And we organized a small group of colleagues and nevertheless friends. And we went to spend a few days in the Lala 2 field station to try to make sense of this data. And we also, once we had considered that it was quite interesting what we were seeing, we invited a few more senior colleagues to participate and help us a bit with their experience, their expertise, and to interpret what we were seeing. And we were quite lucky because we found results that were so cool that they were accepted. They were published eventually in Nature. So essentially what we discovered is that when you combine these 10 traits together, the 10 traits, the 6 above-ground traits and these 4 fine root traits, the resulting unified plant functional space has 4 dimensions of variation. And the most interesting thing is that these 4 dimensions are almost exactly the same as the dimensions of the individual, global spectrum, and root economic space. So the main message here is that the above-ground and below-ground parts of plants are decoupled or not coupled very strongly, let's say, so that the way that a plant looks above ground doesn't tell us much about how the plant is below ground. And this is very interesting because it means that plants have a lot of room, right, to combine different functional strategies in different organs. So a plant can be very small or it can be huge, but this is unrelated to what kind of leaves it has or what type of roots it has, which might be one of the reasons that the diversity of form and function in plants is so extraordinary in the world. So I think this is a very, very interesting, very cool result. And as I mentioned before, the same approach can be applied to any organisms where you can measure traits. And I want to show very briefly an example of something else that we have been working in lately with Dr. Eleonora Beccari, who defended her thesis a couple of weeks ago, or 3 weeks ago already, or 4. So essentially, Eleonora compiled a trait data set for global mammals, and she described a functional space that is also two-dimensional. And one of these dimensions is related to the pace of life of mammal species, and another one is related to the reproductive strategies. And she demonstrated that mammals are quite strongly segregated in that space, depending on whether they live on the ground, whether they live in water, or whether they are able to fly. So Eleonora is around, so if anybody is interested in this particular aspect, you can ask her later, or you can read the paper, which is very interesting too, I think. But let's go back to plants. So once we have defined this space, then we can use it as a map where we can place any vascular plant based on its traits. And this allows us to characterize plant ecological strategies using a standardized and comprehensive approach. So if we have the 10 traits for any plant species, we can put it in this space. We have an example here with 2 species. One is Trifolium pratense, a clover, and the other one is Pinus sylvestris, the common pine that you have here in Estonia. And as you can see, and as everybody, I think, might expect, these 2 plants are very far from each other in the above-ground part of the functional space. But surprisingly, they are quite close in the below-ground part, so their fine roots are rather similar, which is, I think, something that you wouldn't expect to begin with. So now, for simplicity, let's focus only on the above-ground space, and the idea is that once we have information for a lot of species, we can group them in communities. So we can sample a vegetation plot in the field, we can identify which species are there, and if we have trait information for those species, we can put them in this functional space. So for example, here, each point is a species in a Mediterranean community, like the one in the picture, and well, incidentally, these Mediterranean communities are the systems where I did my thesis, and I am quite fond of them, as I am fond of my supervisors, which are here, Begoña and Paco. But anyway, once we have placed species in this space, we can characterize its functional structure, the functional structure of the community, which represents how species in the community occupy the functional space. So the problem here is that functional structure includes all aspects of trait variation. It's not as easy as counting the number of species, which is what is generally done with taxonomic diversity. There are lots of things that we can define here. So it's complex, and our tools are not powerful enough to grasp this full complexity. So what we do is that we generally study individual components of functional structure, like the mean of the trait values, right? The variance, what is the dispersion of the traits around the center of gravity of this space occupied by this community, or the amount of space occupied by the community. But in essence, when we do this, in a way we are acting a bit like the blind man of the parable, who were asked to describe an elephant by touching different parts of his body, right? And depending on what part you are touching, you will describe the elephant in a very different way. So even if we do our best later on to try to reconcile these disparate aspects that each component needs trait capture, we might be missing many important points. And this is why in the last years I have been developing different tools to assess the functional structure of ecological communities, and we have published a few papers applying these tools across different organisms. So the solution that I came up with is to avoid breaking down functional structure and instead trying to use all the information at once. And this can be done with this probability density framework, or TPD, that I have developed along with other colleagues. And the idea behind it is very simple, is to express functional structure as a mathematical function that reflects how the species in a community feel the functional space, so that this function incorporates all the functional structure at once. Well, this is something that I developed mostly while I was in Czech Republic, working with Francesco De Velo and Jan Leps, and also with a lot of help from Norman Mason, who is in New Zealand and a very, very, very cool guy and very clever. And one of the most interesting aspects of this approach is that we can calculate TPD function at any level of organization at which we can measure traits. So this goes from within individuals to the whole world, and we can compare the TPD of lower levels to get the TPD. We can combine the TPD of lower levels to get the TPD of more complex level. For example, in this figure, we start with the traits of a few individuals of different species. Each color represents a species, and if we can measure the traits, then we can do a bit of mathematical magic that I will not explain much here, but we can build some small probability distribution around each of these measurements, and then we can aggregate them to get the probabilistic distribution of a species. And this TPD of the species represents how common each trait value is in this species. So in this particular blue species, you have a lot of, you have a lot of individuals that have these trait values, but you don't have individuals that have very, very high trait values. And the same with all other species. And then the idea is that you can compound these distributions together. So if you have a community that have 3 different species, you can, if you want, you can weight the function of each species in proportion to its relative abundance, and then you can combine them to get the trait probability density of the community. So the idea is very simple, but it's very powerful because it allows us to calculate functional diversity at any spatial scale in a seamless way, right? So one interesting aspect of this TPD function is that they can be compared directly between them because they are probability density function and they have a series of properties that allow us to compare them through how much they overlap. The overlap between these two functions is this area in red, and then we can use this to estimate how dissimilar they are. Essentially, 1 minus the overlap is telling us how dissimilar the trait probability density function of two species, two communities, two countries are. We have used this a lot. For example, here in the mammals paper that I was showing before, Eleonora used this approach to calculate how dissimilar the set of mammals from aerial, aquatic, and terrestrial realms are from each other. So you can see, well, that they are in general quite dissimilar. Here you have in purple the terrestrial and in blue the aquatic, and you can see that they have a dissimilarity of 0.69, but the terrestrial and the aerial are more dissimilar between them than the aquatic and the terrestrial. Okay, and the aquatic and aerial are also more dissimilar. They have a dissimilarity of 0.86. Similarly, the method allows to estimate the functional redundancy of assemblages. The idea with redundancy is that if you have a community where you have many species with similar traits, then the community is highly redundant. And this means that if the community loses a species, its functional structure is unlikely to be very strongly affected by losing that species when that species goes locally extinct. And on the other hand, if a community has low redundancy, all the species are, let's say, functionally unique, then it is likely that the community is going to lose some trait values that were exclusive of one species if this species goes extinct. So its functional structure, let's say, is more vulnerable to extinctions. And essentially for this is very simple. We simply count how many species display each trait value of combination of trait values in the community, and then you can average that across the full community to get a value for the community, but you can also estimate redundancy within individual parts of the space. And this version of the approach, we discovered using this that a large proportion of the functional space occupied by plants and different groups of vertebrates at the global scale is occupied by only one or a few species. So this means that if these species with very unique traits go extinct, then we will completely lose these trait combinations from Earth. So, for example, the case of freshwater fish is very, very extreme. Around 40% of the global functional space is occupied by 5 or fewer species. So it's highly vulnerable to future extinctions. And this means that it's very important to be able to identify which are those species. And this can be done by taking advantage of the fact that we can compare the TPD functions not only between members of the same organization level, but also across different organization levels. So, for example, if we compare the TPD of a species with the TPD of the community in which the species is, we have a good indicator of how unique the species is in the context of that community. And this can be done at any scale, so we can get indications of uniqueness about species within communities, but also species within regions, or communities in regions, or species at the global scale. And this comparison of species, estimation of uniqueness of species at the global scale is something that Kerry Stewart, who's a PhD student that I co-supervise in UK, is working in. She is trying to enhance these indices, and she has found that conserving the top 100 most unique threatened bird species at global scale, if you conserve these 100 species and you keep them from going extinct, then it might be possible to prevent more than 2/3 of the projected losses of global functional diversity of birds in the next 100 years. So I think it's a very interesting tool to try to guide conservation efforts, because of course this is at the global scale, but it can be applied also on national scale, regional scale, any scale that you want. And indeed, we can still estimate individual aspects of functional structure, but using a common framework. So these traditional indicators are essentially functional richness, which tell us about how much space is occupied by an assemblage. So we have the blue assemblage that occupies a lot of space, and this orange assemblage that occupies very little space. We have functional evenness, which tell us about how evenly the traits are distributed within the space, or functional divergence, which tell us about how close to the boundaries of the distribution the peaks of the abundance are. And we have used also these components in many studies. For example, here, in this paper, we show that agricultural intensification at the field scale, so how much fertilizer or how much herbicide is applied in a field, is an important driver of changes in the functional structure of plant communities across Europe. So if anybody is interested in these approaches, you can read the papers, you can talk to me. I'm around and I'm approachable, I think. And in these papers, we have been developing these methods over years. Or you can use yourself a couple of our packages that I have developed with the help of some colleagues in the last few years too. So these are freely available for anybody to use. So in this part of the lecture, I'm going to show you how some of this research works in which we have applied these approaches. So first, I'm going to deal with a paper where we collected a very large amount of trait data for tens of thousands of species of plants, mammals, birds, reptiles, amphibians, and freshwater fish. And we had different goals, but the main thing that we wanted to explore here is how the functional structure of these groups would be affected by the future extinction of threatened species. But for that first, we had to define a functional space for each of these groups. And we already found something interesting that you more or less know about already, because I have described already the global spectrum of plant form and function and the mammal space that Eleonora did. But essentially, across all groups, we found that most spaces were two-dimensional, and the spaces for vertebrates were all fundamentally very different, similarly with a dimension related to the pace of life and a dimension related to reproductive strategies of species. And after defining these spaces, we mapped the risk of extinction in these spaces. And for this, we collected information about the threat classification of a species from IUCN, and we modeled the probability of a species being threatened as a function of its position in the trait space. And we found another very interesting result, which is that almost across all groups, the species that have a slower pace of life or that reproduce more slowly, or both, are much more likely to be threatened with extinction than fast species that reproduce more profusely. In some cases, the threat of extinction for species with certain traits is shockingly high. As you can see, for example, in this area with mammals, this is the space of mammals. In this area, this blue area, almost 80% of the species that have this combination of traits are threatened nowadays. And in the case of plants, it doesn't look so spectacular, but it's still quite substantial. You can see that this area, which is occupied by large trees, the probability of extinction is up to 3 times higher than the probability of extinction of annual species that are around this area. And finally, we explore how these potential extinctions in the future will reorganize these functional spaces, and this is what we call this erosion of the functional space. So these maps show in brown some parts of the functional space that will be more negatively affected by extinctions, meaning that those trait combinations will become much less common in the future. And you can see even some black areas in some of the spaces that represent traits that are likely to go completely extinct from Earth in the next few years. And by contrast, we have blue areas, like this one, that indicate that these species with these traits are much less, proportionally much less affected by extinctions. And here I have a small animation, I hope it works, showing how the functional space of mammals is likely to change if the species that are currently threatened with extinction actually would go extinct. I don't know if I can make it work. Yes. So as you can see, some part occupied by rodents are very likely to remain stable, but other parts, particularly the area to the bottom right occupied by primates, will experience a very significant erosion in the next few years. So in fact, currently, if we go to the beginning of the animation, we have like 3 hotspots of mammal diversity at the global scale, but the one that is occupied by primates is likely to be eroded very, very significantly. Then in this other study, we explore how plant diversity has changed in the last 14,000 years in Northern Europe, but looking at both observed diversity and dark diversity. And we found that the species in dark diversity are those, species in dark diversity are those that could be present locally because their traits match the local environment, but they are absent. And using pollen data, we found that diversity increased significantly after the Ice Age, particularly due to climate warming and human impacts like agriculture. And interestingly, many species delays in colonizing suitable habitats, especially plants with fast life history traits and small size and later seeds, they were slower in colonizing these habitats. And on the other hand, we found that functional dark diversity is generally very small, so that when new parts of the functional space become available after environmental change, then these parts are filled very fast. And this is another study where we examine with Roy from Colombia how different tree species in tropical forests respond to extreme drought, focusing on the 2015 El Niño event in South America. And we analyzed for this trait data for more than 20,000 trees and describe a functional space that show 2 main dimensions. One is hydraulic safety efficiency dimension, and the other dimension reflects investment in plant tissues, so dense wood versus lighter wood. And we found that dominant species make generally high investment in tissues, but they can be either safe or efficient in terms of their hydraulic strategy. And then we cross this trait information with demographic information for those trees, and we found that these 2 main strategies are indeed different in how they responded to this extreme drought. And to explore this, we represented the same space, but when we calculated the TPD functions, we weighted each species according to how much biomass they produce in the sample interval, which is growth, or how much biomass was the biomass of the new recruits of the species, which is recruitment, or how much was the biomass of the individuals that die between the 2 intervals. And we found that both, while recruitment was small in general, hydraulic safe species were much better at it than hydraulic efficient ones, probably because the drought was too harsh for the efficient species, which depend on high water availability. But on the other hand, and this was surprising, hydraulic safe species experience substantially higher mortality during the drought despite they are presumed to be more tolerant to drought, which suggests that the drought severity was higher than the safety margins for these species. So altogether, neither of these species were fully equipped to fight against such an extreme drought, and this suggests that if such droughts become more and more common, then these tropical systems might change dramatically, because new species will come to substitute the species that are currently there. Here we have another example from Enrico, a postdoc who's also working with us, and where he integrated taxonomic, functional, and phylogenetic dimension into a single metric called mu diversity. And essentially, mu diversity takes the number of species in an area and it weighs this number according to how redundant these species are in terms of traits and in terms of their evolutionary history. So it represents not just how many species are present, but also how unique these species are functionally and phylogenetically. And it provides a metric that should be closer to true ecological diversity. And his results highlighted that some regions like Papuasia, Southeast Asia, Australia, and Central America are hotspots with excessively high and unique mu diversity. And then we explore what are the main drivers of this mu diversity. And we found that historical climate stability and current water energy availability are the most influential factors. But interestingly, we also observed that the legacy of early human activities, like the onset of pastoralism, still influences plant diversity today very strongly, underscoring that both past and present human actions influence plant diversity very much. And another one from Eleonora. In this one, we were interested in examining if plant spectral diversity obtained from high-resolution multispectral images can capture patterns of functional diversity in coastal dune plant communities. And this is important because if remote sensing can detect biodiversity changes, then it provides a fast and cost-effective approach to monitor biodiversity. So they sample plant communities along different transects in coastal dune ecosystems in Italy, and in each transect they have multiple small plots where they measure species present and the traits of species, and they also took images with a drone. And then we apply this functional space approach to the traits of species, so we have traditional functional space based on traits, but we also made a spectral space. In this case, the different bands of the images are the traits, and each pixel could be considered as a species. And we found that there is a strong correspondence between spectral and functional diversity, suggesting that the spectral diversity closely mirrors ecological trait variation. And interestingly, spectral diversity did not reflect taxonomic diversity. So it was very weakly correlated to the number of species, to species richness. So we underscore the potential of remote sensing to measure functional diversity, but perhaps not so much taxonomic diversity. And to finish this overview of our recent research, I want to mention a couple of people who have been very important in the last years also in my group, who have focused mostly on using the same approaches but in experimental settings. And I will go not much into details of these papers, but I also wanted to acknowledge Slendy and Jean. And finally, I'm going to show what we are currently trying to achieve very briefly. And the idea is that now that we have developed and we are in our way to master these tools that I have presented before, I would like to really tackle this response effect framework. And for that, the main barrier is that there are very few species with enough trait information to make this space operational, particularly for the fine root part. So to solve this problem, or try to solve it in the best way that we can, we have established a global sampling collaboration to characterize the traits of a very large amount of species. And this collaboration is called TraitIfNet. We coordinate it here from Tartu. And as you see, we have a lot of people involved in TradingNet. And in TradingNet, we already have more than 200 groups of researchers from all continents that have joined and are measuring trades in their own systems. And in addition to this, we are organizing our own sampling expedition, thanks to funding that we have recently acquired, where we try to measure traits of key species and ecosystems that are not well represented by the participants involved in TradingNet. And the idea is that combining this trait information with information about the species composition from plot databases, we can characterize the functional structure of hundreds of thousands of vegetation plots across the world. And then, this way we can link the TPD function of each plot with its environmental variables, with different indicators of ecosystem functioning, and then we can use this information to model the relationship between environment, functional structure, and ecosystem functioning. And for this, I plan to use deep learning approaches that are very good at generating images, so that they will, if this approach works, it will learn what are the rules behind these arrows that link the three components. And hopefully, once we understand these rules, we can try to apply them to different global change scenarios to try to predict the future of biodiversity from functional traits. So hopefully, we will be a bit closer to this promise that I think is still unachievable, but is a good goal to have, of this holy grail of ecology. And here I finish my lecture. Thank you very much.
Thank you, Carlos. Now it is possible to ask questions, comment. Yeah.
Thank you for the talk. In that animation that you showed in the slide, I think you were showing the erosion of functional diversity or something like that. Mm-hmm. And you mentioned that there were two hotspots that tend to be stable through time. Yeah.
Not through time. I mean, this is present time and how the near future is going to be. Mm-hmm. If the species that are threatened with extinction are going to change. This doesn't represent changes like evolutionary in evolutionary time. Mm-hmm. Yeah, mm-hmm. It's like, let's say, how the functional space of mammals is going to change in the next few hundred years if the species that are threatened go extinct.
Mm-hmm. And do you have a reason for the stability of these two hotspots? I was wondering if it would be due to the fact that these are the most diverse groups within mammals.
Yeah, you could think that, but as you can see, if you see, and let me see if I'm able to show this without starting the animation. There are these areas with a thicker black line that represent the 50% quantile of this distribution. So these are what we call the hotspots. And as you can see, there are actually three hotspots. There is one here, which is this one occupied by primates. And the thing is that primates are much more threatened with extinction than the species in these other two hotspots. So it's not that the hotspots are stable per se, because if the species that are in a hotspot are disproportionately threatened, then the hotspot is going to be eroded very much. The idea here is that the traits that are associated with extinction risk are different, so that the particular species that are in this hotspot and this hotspot are not so much currently threatened on average. Of course, you have threatened species everywhere, but the species that are in this hotspot, in this area, they are much more predominantly threatened. So this is what we wanted to show, yeah.
Okay, Some new questions? Yeah?
So thank you very much indeed. You had this nice result that when you addressed above ground traits, you had these 2 centers of density, and below ground you have 1. Mm-hmm. When we have studied fungi, then we have repeatedly had a case that we find nice pattern, and when we collect more data, it will not become more clear, but it will disappear. Mm-hmm. And here the root data are available about so few plants, and if you think how many plant species are on the globe, can't it be just the matter of too small sample?
It could be. I think it's not, but it could be. I mean, we have 300 species in this Nature paper, which is, I think, like 0.1% of the species that are plant species that are estimated to be in the world. Very, very, very little. And this is why we have started this TradeWithNet initiative to try to collect data for many, many more species. Right now, I don't have the number in my head, but we have already in Tartu samples and traits for more than 1,500 species, in addition to the 300 that we have. So we will be able to see if this is true or not, and if the pattern become more interesting, or it continues being not very well defined for fine roots. But in the experiments that we do, and like the experiments that Jean is doing, and the experiments that Slendy has done, and so on, we tend to find the same thing, like very little differentiation between species in their fine root traits. But which is also interesting, because we find much higher differentiation within a species in the fine root traits. So it looks like, I don't know exactly why, but somewhat the individuals or the roots of the different species, they are able to be much more different than the aerial parts, somewhat. So this is also something that I find very, very interesting to study, but I don't know exactly if it's going to remain when we have many more samples. So whatever we find is going to be very interesting, I think.
Thanks.
Yeah, this is always an interesting question about this data size and what happens. Okay. Yes. Can you please come to microphone?
Yeah, relatedly, I'm thinking about the predictive power of traits. So I get an impression that especially traits related to physiological tolerance and that kind of stuff that might be really important with global change is kind of understudied. Do you agree? And are there other like further blind spots that could have great predictive power that are not really well studied?
Yeah, this is a bit the concept of soft trait and hard trait that has been used very much. So you have soft traits that are somehow easier to measure, but perhaps not so informative, not so close to performance of plants. And then you have traits that are very close to the performance of plants, not to the growth, to the reproduction, to the survival. And these generally are more physiological traits, and they are harder to measure. You have to go and do complicated measurements, spend a very long time measuring photosynthesis or thermal resistance of a species, no? At what temperature a species dies, and so on. So yes, of course, there are many traits that are much more mechanistic somehow, but at the same time they are much harder to measure. And since I am mostly working at macroecological scales, that makes it a bit problematic, because I don't have any hope that these traits are going to be defined for many species at any time soon. But at the same time, the traits are kind of all somewhat related to each other, so you can use some other proxy traits, like specifically Varia, which is related to photosynthesis. It is a weaker link, but it's still there. And this is more or less the approach that we are following. But of course, the ideal would be to measure the really mechanistic physiological trait. I agree.
Thanks. Okay. More comments? Yeah. Mari?
Thank you, Carlos. I just sitting here and thinking about the possibility, and it will be a provocation now. I will provoke you. You know that I'm also working with DNA data, and I think it's the most convenient way to study large-scale, let's say, landscapes, the globe, because it's very easy to compare different places, different communities with different, with the same tools. Mm-hmm. And I know that maybe a decade ago or even 20 years ago, when this trait-based ecology started, then there was an attempt to predict traits based on phylogeny. Mm-hmm. That the traits and they are very phylogenetically related species which are more close to each other, they behave more similarly, or the traits are more similar, which is in general probably it's true, mm-hmm. but it's not so well established. Well, it doesn't really, it's not the case in plant kingdom, mm-hmm. I would say. Mm-hmm. So, but basically, do you see in the future possibility to draw those fried eggs or trait spaces based on DNA data, based on molecular analysis and molecular species? It's okay. Just...
So you mean without previously having measured the traits?
Yes, okay. Without really measuring things. It's just you go out, mm-hmm. sample DNA from environment, and have your species list, and then... or gene list.
Yeah, what we do is partly like that, but we have to measure the traits first. So if you get the DNA and you can identify the species, then you can, if you know where they are in the space, then you can add them. But what you mean is a bit different, and I don't think that's very possible, because it depends a lot on the group. We are seeing this because we are trying also to see how reliable are approaches to impute the missing trait values of a species, right? And these approaches are based on the correlation of traits. So you have measured some traits of one species, but you don't know another traits, then you can try to infer from the traits that you know how are the traits that you are missing. Right, and phylogeny in general is supposed to be very helpful in this, because if I tell you, okay, this species has a height of 20 meters, perhaps you cannot tell much about specifically Varia. But if I tell you it has a height of 20 meters and is a conifer, then you know the specific area is going to be small, because it's a conifer and they have thick leaves, right? But it doesn't work in all families. So you have families like, I don't know, in plants like the legumes that have very small herbaceous plants and very big, huge trees, right? So I think there is some hope to do that, but I wouldn't put all my hopes in phylogeny, to be honest. But on the other hand, we are seeing that with mammals, for example, and the traits that we are using, these are much more strongly conserved, actually. So it depends a lot on the group, but plants are very diverse, let's say, within the same area of the phylogeny. The plants can be really very, very different with these traits that we are considering, yes.
Mm-hmm. Okay. So if there is no any questions, then it is possible to congratulate our professor in case of inauguration lecture. you are all free to go and congratulate. And then there are some snacks in the University Art Museum, which is on the base floor of this building. So don't miss that.